Very good morning. We are from Group 14 and our group name is The Breath. I'm the group leader, Muhammad Akmal, and my friends will follow the introduction.
I'm Listen Darius.
Hi, I'm Chinwan. I'm Chanjuan.
Essentially, there are four big subtopics for the respiratory system. First one is the main function of the conducting portion, the structure of the conducting portion, the specialized cells of the alveoli, and the structure of the respiratory portion. As you can see in the video, we'll go deeper into each subtopics.
And now we'll be talking about the conducting portion. Basically, conducting portion has two main functions, which is to provide the conduct through which air can travel to and from the lungs, and to condition the inspired airs. To ensure an uninterrupted supply of air, a combination of cartilage, elastic fibers, and smooth muscles provide rigid structural support and flexibility and extensibility to the conducting portion. Cartilage is found in periphery of lamina propria. It is to support walls of the conducting portion, thus preventing collapse of lumen and ensuring the continuous supply of air. Next, elastic fibers provide flexibility. We can say that the concentration of elastic fibers is inversely proportional to the diameter of the conducting tubules. And while smooth muscles are found encircling on the tubes from trachea to alveolar duct, the contraction of the smooth muscle will reduce the diameter of the conducting tubule and regulates the airflowing. As I mentioned before, the function of conducting portion is to condition the air and inspired air. To do this, we can find vibrissae, which is to remove the particles of dust and other substance, and mucus and serous secretion, which is to moisten incoming air and protect the delicate alveoli from desiccation, while capillaries we can find also to warm the incoming air. To move on to the respiratory epithelium, most of the portion of conducting portion is ciliated to the stratified columnar epithelium and containing rich population of goblet cell, and it is called respiratory epithelium. A typical respiratory epithelium normally consists of 5 types of cells. The first, it's ciliated columnar cell, where it is the most abundant type. It has about 300 cilia on its apical surface, and mitochondria can be found beneath the cilia to supply ATP for ciliary beating. Second, it's mucous goblet cells, where it is the next most abundant type of cells. Its apical cytoplasm contains mucin granules. Third is brush cells. It is called brush cells because it contains numerous microvilli on its apical surface. It is a columnar epithelial and have afferent nerve ending as basiles on basal surface that serve as sensory receptors. Fourth, it's basal or short cells. They lie on lamina but do not extend to the luminal surface of the respiratory epithelium. They are small rounded cells, and they are generative stem cells that undergo mitosis and then differentiate into other cell type. Fifth is the small granule cell. It resembles a basal cell. It contains numerous granules, which is to control the secretory activity of goblet cells and secretory gland. In the respiratory epithelium, we can also find a type of non-keratinized stratified squamous epithelium from the nasal cavity through the larynx. They are found in regions exposed to direct airflow or physical abrasion, for example, oropharynx, epiglottis, and vocal folds. The stratified squamous epithelium provides protection from attrition more than the respiratory epithelium.
So I'll be talking about the structure of the conducting portion. They are divided into 4. There is the trachea, bronchioles, bronchi, and also the bronchial tree. For the trachea, they extend from the larynx, and they are lined with the respiratory mucosa that consists of the respiratory epithelium, thick basement membrane, and also the supporting lamina propria.
The lamina propria are also present, and they consist of about 16 to 20 C-shaped rings. And the gap between each incomplete ring is bridged by the smooth muscle, trachealis muscle, and also the fibroelastic ligament. Okay, now I'll go into bronchioles. They don't have any cartilage and glands, and they also have scattered goblet cells in epithelium and also the large bronchi. In the smaller bronchioles, they are ciliated with the simple columnar and also cuboidal cells that line it without the goblet cells. In the terminal bronchioles, pleural cells is also present. For the apical part, they contain secretory granules that secrete proteins that protect the lining of the bronchioles. And the lamina propria is also present, that consists mainly of the smooth muscle and the four and also elastic fibers. In the large bronchioles, it is lined with the ciliated pseudosatisfied columnar epithelium. Now I'll talk about the bronchi. The bronchi, the mucosa is structurally similar to the mucosa of trachea, except that the organization of the cartilage and the smooth muscle is different. While for the bronchial cartilage, they are more irregular, and in the large bronchi, the cartilage rings are completely encircling the lumen. For bronchial tree, they divide into two primary bronchi, and they subdivide more into three secondary in the right and two in the left. For the secondary bronchus, they supply a pulmonary lobe that is called the lobar bronchus. For the bronchus, they divide repeatedly to form smaller bronchioles that houses the bronchioles at the end, and the bronchioles branches to form the terminal bronchioles.
So now we are going to discuss about the structure of respiratory portion. Respiratory portion divided into three parts, which is respiratory bronchioles, alveolar duct, and alveoli. For the respiratory bronchioles, it basically serves as a transitional between conducting portion and respiratory portion. Numerous alveoli extends from the wall of the respiratory bronchioles. Some parts lie with the ciliated cuboidal cells and pleural cells. In the more distal part of the respiratory bronchioles, the cuboidal cells there do not have cilia, and we cannot find goblet cells here too. Lamina propria of the respiratory bronchioles consists of smooth muscles and elastic fibers, and it is terminated by branching into alveolar duct. Now we are moving to the alveolar duct. Alveolar duct is thin wall lined with simple squamous epithelium. The lamina propria of alveolar duct consists of smooth muscle, elastic fiber, and collagen fibers. It leads into numerous single alveoli and alveolar sacs. There is elastic reticular fibers form a complex network encircling the opening of alveoli, alveolar, and alveolar sacs. The functions of the elastic fibers is to enable alveoli to expand during inspiration and contract during expiration, while the function of reticular fibers is to prevent overdistension of alveoli. For the alveoli, it is responsible for the spongy structures of the lungs. Oxygen and carbon dioxide are exchanged between air and blood here. The structure of the alveolar wall is specialized for enhancing diffusion between external and internal environment, because alveolar wall is one cell thick, and then it is moist and surrounded by network of capillaries. Each alveolar wall lies between two neighboring alveoli. Alveolar wall can be termed as interalveolar septum or interalveolar wall. The interalveolar septum consists of two thin squamous epithelium layers. Between is the interstitium, and we can find capillaries, connective tissue, and fibroblasts here. So now I'm going to discuss about the blood-air barrier. Air in the alveoli is separated from capillary blood by blood-air barrier. These three components consist in the blood-air barrier, which is cytoplasm of epithelial cell, fused basal lamina of epithelial and endothelial cells, and the cytoplasm of endothelial cells. Oxygen passes into blood through this barrier, while carbon dioxide diffuses in the opposite direction. The total thickness of blood-air barrier is about 0.1 until 1.5 micrometer.
Specialized cells of the alveolar. Firstly is the capillary endothelial cell that lines the capillaries in a continuous way and thus have no fenestrate. And secondly is the alveolar epithelium that comprises of type 1 cell known as squamous alveolar cell and type 2 cell known as the septal alveolar cell. The type 1 cell it has occluding junctions with desmosomes to prevent leakage of the fluid into the alveolar air space. Besides, it also has a large area of free areas in the cytoplasm where the organelles are grouped around the nuclei in order to reduce the blood-air barrier flow, and the main role of the type 1 cell is to provide barrier of minimum thickness to the alveolar surface. Next, the type 2 cell, they are distinguishable from the type 1 cell with their larger nuclei and more rounded shape, and also their foamy appearance due to the presence of granules. Their lamellar bodies discharge their content over the surface of the alveolar to form a thin layer which contains the pulmonary surfactant that reduces the surface tension and also prevent the alveolus from collapsing. Finally, the type 2 cell also carries out mitotic division to replace its own population and the type 1 cell. Moving on to the alveolar macrophages, also known as dust cell, they clean the epithelial cells by phagocytosis, and they are found in the interalveolar septum and also the alveolar surface. So for the macrophages that is filled with dust or other particles, they will be passed through the alveolar to the bronchioles by amoeboid movement, and then to the pharynx with a layer of mucus where it is swallowed. There is alveolar pores. Alveolar pores are located in the interalveolar septum to connect the neighboring alveolar cells. The function of alveolar pores is to equalize air pressure in the alveolar space and also to provide alternative route for air movement when there is obstruction. That's all. Thank you.